All right, so we're going to get started here with the genetics notes for the monohybrid cross and Punnett squares. So we're going to start off with our learning target for today. So students will be able to calculate the probability of offspring having specific genotypes, phenotypes, given the parental genotypes. So based off the parents, we are going to be able to tell the genotypes and the phenotypes of the progeny or the children of the cross that we do. All right, so our learning targets, we want you to be able to express probabilities using phenotype and genotype ratios. We want you guys to be able to express probabilities using the percents and fractions, and then we want you guys to be able to calculate probabilities using a monohybrid Punnett square. All right, so a Punnett square is just going to be a grid system for predicting all possible genotypes resulting from a mating or a cross. So when we talk about matings, we're going to be talking about a cross, and the two alleles from each parent are positioned on the axes and then crossed. So it's the most important thing about the Punnett square is how we get it all set up. So for example, if we had R, the dominant, so we have big R, little r, and the big R is going to stand for able to roll your tongue, and the little r is unable to roll your tongue. So if we were given that the father is homozygous recessive and that the mother is homodominant, we would set it up so that the father's genes are on the top here. So little r here, and then the mother's genes are going to be big R, big R. So the way that I got that was if we are going to do the mom here, and we're crossing it with the dad, we know that since the mother is homozygous dominant, that she can only have alleles for big R, and that since the father is homozygous recessive, can only be little r. So the only alleles that the dad can give are going to be the sperms are going to all have little r in them, and then the mom can only give big R in her egg cells. All right, so the monohybrid cross, which we are specifically looking at today, is going to be a cross that examines the inheritance of only one specific trait. So over here we have peas, and those peas can either be green, which looks like it's going to be dominant. So green is dominant. And then we also have yellow, which it looks like is going to be recessive. And we can tell that just based off the fact that the big G is present. We get green pea pods and not the yellow. The yellow only shows up when there are two little g's present. So probability is just going to be the likelihood that a particular event will happen. It is a predictor for the average number of occurrences and not the exact number. So, for example, over here we have a penny and a nickel being flipped. So if we flipped the penny and nickel, we would have 4 possibilities. We could either get heads with the penny and heads with the nickel. We get tails with the penny and heads with the nickel. Get heads with the penny and tails with the nickel. Or we could get 2 tails. So the probability of getting 2 heads here is 25%. The probability of getting 1 heads and 1 tails is going to be 50%. And the probability of getting 2 tails is again 25%. So the probability will be the number of ways a specific event can occur. So in this case, to get a tails and a heads, there would be 2 total ways for you to get that. Or there would be 2 numbers of ways that that specific event can occur. And then for the number of total possible outcomes, there are 4. And that's how we get that 1/2 or 50%. Okay, so Mendel's experiments. So Mendel began experimenting with purple flowered pea plants. With white and purple flowered pea plants, excuse me. So he crossed a homozygous white and a homozygous purple pea plant, which resulted in all purple flowers. And he wanted to know where that white allele went. So one thing that I want to point out is that we can already tell that the purple is going to be dominant. And we can tell that simply because when we crossed a homozygous purple with a homozygous white, the only thing we saw was going to be the purple. So to find out where that white allele went, Mendel did a self-cross with the plants. So that just means that he basically took each plant and had it fertilize itself. The result of that was that there was about 1 white flowering pea plant to every 3 purple flowering pea plants. You can see that over here. When he crossed them, you got a pea here, or you got the purple here, purple expressed here, purple expressed here, and then white was only expressed in the 1. Okay, and what does that mean? So it means that one gene is going to mask the other, which just proves that one allele has dominance over the other allele, because this white was not being crossed at all, or was not being expressed at all in that initial cross. So what we call these 2 that were used right away, so we call that the parental generation. For all of these that are in the cross itself, in the Punnett square, this is going to be the F1 generation. The F in F1 stands for filial. And then, so from there, we then self-crossed those plants. And this would just be the F1 generation again. And then the F2. And this important thing about the F2 generation is that that is where the white allele was expressed once again. Because remember, it wasn't expressed at all in the F1. There was 0% chance for the white allele to be expressed in F1, and about a 25% chance for it to be expressed in the F2 generation. Okay, the law of segregation. So Mendel concluded 3 things from his observations. We're going to look at 2 of those things. And the first 2 are called the law of segregation. So the first part of the law of segregation is that organisms inherit 2 chromosomes and therefore 2 alleles. So 1 allele from each parent. The second part of the law of segregation is that organisms can only donate 1 copy of each gene into a gamete. And we know that already from meiosis. So we know that the mom will only contribute 1 of her genes to a gamete, and a father will contribute 1 of his genes to a gamete. And then when those come together, they will form a diploid cell that can give rise to a potential offspring. So right up here is going to be that the law of segregation. So we're going to start out with the full number of chromosomes present in the cell, and in this case that's going to be 6, and the homologous chromosomes again are going to be the same color, much like we in the meiosis modeling activity that we had done. So as you can see here, this is going to be the diploid number for all somatic cells. So 6 is the diploid number for this particular species or organism, and it is the somatic cells that contain that diploid number. So the possible gametes, which are shown down here, remember gametes are going to be the sex cells, and these potential or possible gametes are going to be haploid. So they have half of the number of chromosomes, so 3. So there are no homologous chromosomes in the haploid cells. All right, and then we'll get into a test cross, and this is a very specific type of cross, and it's between an organism with an unknown genotype and an organism that is homozygous recessive. So the reason that it's important to always go with the homozygous recessive trait in the test cross is because you'll always know that it's homozygous recessive because of its unique expression. The reason that you might have an unknown genotype is because it could be big P or big P little p. So it could be either homozygous dominant or heterozygous. And that's also why we don't want to use any of the, we don't want to use a homozygous dominant or a heterozygote because we might not even know what that is. And if you were to cross a big P and a big P here and a big P and a big P here, you would always end up with purple in both instances. So it's important that you use the homozygous recessive. So if you do the cross with a big P, or in this case, the homozygous dominant, all of the progeny will be purple because they will end up as heterozygotes. All of the progeny will end up as heterozygotes, which is exactly what happened to Mendel in his first experiments. So this resembles a lot like the F1 generation from his experiments. If you have a heterozygote crossed with a homozygous recessive, you're going to get half purple and half white. So there's a 50-50 chance on either of them. All right, and that concludes the notes for this unit. Make sure to come with any questions, and thank you for listening.